So your final exam is on Thursday, right?
Wednesday.
Okay, Wednesday. In here? I presume it's in here, yes?
Better be, because I'll be here and somebody else will be here.
That final exam will be very similar to the last exam. It's going to be obviously be on the most recent material, which is primarily, there's a little bit of motor system tracks, I think, you know, so Parkinson's onwards. Then it was cerebral blood flow, autonomic nervous system, and then a bit of reticular formation. We'll finish off the limbic system today. So not much on there really. It's less percent-wise than the previous exam. So I know you've got a lot of other exams, so it's very hard to study this time of year. It'll be, you know, I don't know, the usual 45 multiple guess kind of questions and a few short answer. And for the PTs, you'll have about 25 multiple guess questions on your comprehensive final, your mock boards exam. Those 25 questions will be more comprehensive. It won't be just on the latest material. There might be one or two on the latest material, but those questions will cover all the material we've had throughout the semester, and it will be more the more the basic information from, you know, the more important information from earlier sections. Okay, since the OTs don't have that comprehensive final, that sort of imitation boards final, those 25 comprehensive more questions I'll just tack on to the end of your exam, and you can take it at the same time. There's a block scheduled for 2 hours. Normally the exam won't take more than an hour and a half at top, so you should easily have plenty of time to do those extra multiple choice questions with the OTs. If you need extra time, we'll carry on, whatever. Okay? So any questions about that final and comprehensive final exam? Okay? So we'll finish off today finish off the reticular formation, and then do a little bit about the limbic system, and maybe talk about a few lesions, basic lesions. You know, perhaps we mentioned them very, very early in the semester, but we didn't get round to it. No class on Monday, remember. I know the PTs have got exams then. If anyone wants to come and see me any time between now and the final exam, please stop by and I'll try and answer any questions you have. And please have Chris and Jen as well to review, but you can feel free to stop by and email me a question if you like. Okay, quickly overview of what we talked about the reticular formation. 4 major functions: somatic, motor control, musculoskeletal muscles are controlled by the medial and lateral reticular spinal tracts. We have sensory control. It has an influence over our sensations. It has influence over the viscera, or autonomic nervous system, and has influence over our level of consciousness. Remember, the motor control comes out of our medial or pontine reticular spinal tract and our lateral or medullary reticular spinal tract. The pontine reticular spinal tract, remember, facilitates our extensors. So you can think of the pontine pushes. The pontine extends the arms, extends the legs, inhibits the flexors. The opposite is true for the lateral reticular spinal tract, the medullary reticular spinal tract. That's going to facilitate the flexors and inhibit the extensors. So the balance between these in terms of increasing or decreasing flexor extensor tone in the limbs. If you damage this, you might get way too much flexor withdrawal reflexes to unobnoxious stimuli. Normally you would pull your hand away from obnoxious stimuli. Patients with damage to these tracts sometimes will pull their hand away from things that really aren't painful at all. Okay? Remember also that the medullary reticular spinal tract is thought to be one of those tracts important in being the command neurons for our central pattern generators, our locomotor and neuromotor circuits within the cord. The command neuron, to get that circuit kicking around, is thought to be one of these reticular spinal tracts. The reticular formation has influence on our sensations, descending influences. When pain information comes up to the reticular formation, it can send signals back down to pain gate out some of that pain. For example, the raphe spinal tract comes out of the raphe nuclei, goes down the cord, ends on the dorsal grey horn and releases what?
Serotonin.
Serotonin. That kind of, and those inhibitory interneurons to try to stem the amount of painful information coming up to the brain. Autonomic control. Remember, the reticular spinal tracts are the upper motor neurons for the preganglionic neurons within the cord and in the brainstem. Okay? So the parasympathetic and sympathetic neurons, they are initiated by reticular spinal tract. Okay? Which of course the control for that is the hypothalamus. And as we talked today a bit, the amygdala, part of the limbic system, will affect the hypothalamus, which affect the reticular formation, which affect the somatic nervous system. Make sense? So the chain of command from the limbic system to the hypothalamus, hypothalamus to reticular formation down the cord to our preganglionic neurons within the cord to affect changes in the somatic nervous system. Okay? The last thing we mentioned was control of consciousness. The level of wakefulness and alertness is a function of the activity of a reticular activating system, as it's called, another term for it. The reticular formation or reticular activating system. It activates the brain, keeps us awake or puts us to sleep. Damage this, coma or lack of sleep. No, complete insomnia. I mentioned the Glasgow Coma Scale, highly prognostic of the patient's future outcomes. The patient comes in with a very low Glasgow Coma Scale. That's pretty prognostic of how they're going to do. High correlation between their future deficits and abilities of the patient and what they come into the ER with based on their Glasgow Coma Scale. Narcolepsy, people who have uncontrollable sleep attacks, cataplexy, where they have uncontrolled loss of muscle tone completely, they'll just collapse on the floor. Again, I've had this, one student had this weird narcolepsy, and that's, I couldn't believe how many weird symptoms this patient presented with, and he eventually diagnosed it with as narcolepsy. So keep that in the back of your mind when you get something that you don't know what the heck's going on. It looks like fibromyalgia, looks like chronic fatigue syndrome, might be polymyalgia, whatever. One of those things, it might just be narcolepsy. Okay, the last thing we talked about reticular formation, which we haven't mentioned last class, was the reticular formation. Reticular nuclei are centers for production of neurotransmitters. Some important neurotransmitters made within reticular formation and sent around the central nervous system using axonal transport, of course. And there's these four areas shown here, four neurotransmitters anyway. Okay, let's look at a few of these. We know that the substantia nigra produces dopamine. That dopamine is usually limited to the nigrostriatal system. In other words, the basal ganglia, essentially. The dopamine that's produced in the substantia nigra is basically used within the basal ganglia. However, the ventral tegmental area is also a center. You can see it there, and here those are the blue dots at the top, or purple dots. The ventral tegmental areas are important also in the production of dopamine. Okay, you can see where this is sent, the purple lines. It's sent into the prefrontal cortex, into the frontal lobe, into the amygdala and hippocampus, which are, we've got today, part of the limbic system. Okay? And it's thought this is, the frontal lobe here, for example, along with the limbic system, is important in motivation and decision making. The dopamine is important in decision making and as the limbic system in motivation. Interestingly, a patient on cocaine, the ventral tegmental area lights up like crazy, and other sort of drugs. So that is why, for example, somebody on cocaine might decide to jump off a building because they think they can fly, or do silly things like trying to drive a car, when you're high on cocaine because it basically affects the ventral tegmental area, which affects decision-making processes. The decision-making processes are basically whacked if they're really on, too much cocaine will have a major effect on a patient's motivation and their decision-making of what they think they can do. The pedunculopontine area, where is that one here? The pedunculopontine nucleus, you can see it here in the little red dots. This is a major center for the production of acetylcholine. Here it is. Okay, this projects acetylcholine, you can see, into the frontal lobe, thalamus. You can see the arrow going for thalamus there, and cerebellum, and brainstem. That's a spider effect with that. These are showing the pathways of this acetylcholine projected right around the brain there, the lower part of the brain. Very important. I mean, this is where the cerebellum gets its acetylcholine from. These fibers send acetylcholine out to be used by these other parts of the brain. The Rafe nuclei, a third center. The Rafe nuclei, of course, we've seen that here, that big red line down the middle there. The Rafe nuclei, as we saw before, are very important in the production of serotonin. A lot of the serotonin, you can see the widespread connections there, those orange lines going all over the brain. Most of the serotonin used around the brain is going to come from the Rafe nuclei, throughout the limbic system, throughout the cerebellum. Remember, serotonin is important in mood. Prozac, remember, prolongs the availability of serotonin in the synaptic cleft. It prevents the uptake of serotonin. So if a person's clinically depressed, they might be using Prozac to improve, overall mood. Remember also the Rafe spinal tract comes out of these Rafe nuclei, down the cord. That's going to project our serotonin onto our dorsal grey horn nuclei. That's going to prevent pain. That's going to alter pain perception as it coming up the cord. So very important production site for serotonin used throughout the central nervous system is the Rafe nuclei. Last one, there's a couple of areas here that produce norepinephrine. Okay? Remember, norepinephrine is released at the postganglionic neurons of our sympathetic nervous system. You'll also find a bit produced in the adrenal glands. The norepinephrine in the central nervous system up here is being produced by these two areas. The medial reticular zone nuclei and the locus ceruleus. You read that there? The locus ceruleus. You'll see that ceruleus spelled different ways. Sometimes there's an OE in there, sometimes there's an AE in there, CAE, COE. But these areas down here, the locus ceruleus and the medial reticular zone nuclei, right there. Here's the locus ceruleus. Locus obviously means location, a small spot. Ceruleus, blue. That's a blue spot. It looks kind of bluish in a dissection. These produce, this is the source of most of our norepinephrine in the central nervous system. It's produced here and sent out all over the place. You find this, the locus ceruleus is kind of correlated also with the amount of attention, as it were. When somebody's highly cognitively attending an exam, for example, the locus ceruleus is lighting up like crazy. When a person's asleep, it's basically dead. There's no activity in the locus ceruleus when a person's sleeping. When they're highly active, awake, alert, really high attention capacity, you find the locus ceruleus going, lighting up a lot. The locus ceruleus is activated by stress. Under stressful conditions, that thing fires, it lights up. increases norepinephrine secretion. Therefore, it alters cognitive function, particularly in the frontal cortex, where you can see all that norepinephrine being projected into the frontal cortex. Okay, and then down, it's going to also influence our sympathetic via the reticulospinal tracts. Our sympathetic tone, the sympathetic outflow is going to be influenced via stress activating on locus ceruleus and the reticular formation. Okay, that's why, for example, on Friday when the PTs are taking that factory exam, right? Very stressful. You have butterflies in your stomach, and you just forget the basic mobilization or assessment of the shoulder girdle. You sit there and you start sweating, blotch your face, and you just thought processes have gone. You cannot think. The basic, the most basic test for, labral tear, whatever. You know that. But you go into the test, stress level shoots up. A lot of pressure. You've got to pass the exam to graduate or whatever, to go on. Okay? Therefore, the locus ceruleus floods your system with norepinephrine. Brain goes to mush. Okay? It feels like that. Butterflies in the stomach. Sympathetic nervous system firing like crazy. Sweating. Your mouth goes dry. You get that chewy sweat. Not chewy sweat, chewy. Well, I get that too. But you get the chewy saliva. And you can't, you sit there and what on earth am I supposed to do here? 10 minutes earlier, or a day later, with nobody examining you, that's a piece of cake, I know exactly how to assess the AC joint. But under high stress, this system kicks in. It's your flight and fight system. You basically want to run out of the room screaming. But that's what's happening. It's just, you're getting flooded with norepinephrine. Your frontal cortex, your decision making, your cognitive functions goes to pot when you start, if you're under way too much stress. So you've got to, obviously calm yourself down and you'll be able to do the exam piece of cake, right? Or should be anyway. Norepinephrine is also projected down the cord from the locus ceruleus. So we have what's known as a ceruleospinal tract, which I've mentioned before. There's small sort of fibers coming out of the locus ceruleus called the ceruleospinal tract. Those projections also end on the dorsal horn, the dorsal great horn, the segmental level. Okay? And you're going to get release of norepinephrine into the dorsal great horn. Another mechanism of damping down pain and sensations. Another way of gating out, pain gating, some of this obnoxious stimuli coming in. It's a very important formation, the reticular formation. Lots of stuff. Important functions in the brain. Sometimes this is forgotten, how important the production of neurotransmitters are from the reticular formation. There are 4 functions. Remember the 4 functions: the consciousness, motor control, visceral control, sensory control. Okay? Okay, let's move on to the last system to look at, the limbic system. A very brief overview of this, the limbic system. Okay. The limbic system. The limbic system basically is a very old part of the brain. It sits between the diencephalon and the cortex, the cerebral cortex. It's deep within the cerebral cortex, beneath the cerebral cortex, on top of diencephalon, on top of the thalamus and hypothalamus, etc., etc. Okay? Sometimes you can divide the cerebrum itself into 3 layers, based on evolutionary past, as it were, and how recently they've developed. The most advanced part of our brain is the outer layer, the most superficial layer, the top layer. And that's called the neocortex, the new cortex. Primitive animals do not have one of those. Okay? Beneath that, the next sort of layer, going around the corpus callosum, is the mesocortex, the middle cortex. the mesocortex. And that largely comprises the limbic system. The limbic lobe, should I say. The limbic lobe. And even the older part of these three layers, the last layer down here is the archic cortex, the ancient cortex. So you've got the ancient, middle and new cortices, these three sort of rough categorizations or layers of the cerebral cortex. And the most advanced, of course, is the neocortex. The oldest is the archicortex down here, and then here's the limbic lobe here, the mesocortex. The components of the limbic system are different whichever textbook you look at. Unfortunately, like the basal ganglia. But most of people will agree with this, I think, characterization I've made up here. The neocortex, we've talked about that before. The motor cortex, sensory cortex, associated areas, though that's the outer layer, okay? Beneath that, we have our mesocortex, the limbic lobe, okay? And this is made up of, you can see those various gyri, hippocampal gyrus, cingulate and subcallosal gyrus. Those are three gyri making up this limbic lobe, which you can see that grey line above the corpus callosum essentially. Okay? Hippocampal formation is basically archic cortex, and here we have hippocampus, dentate, gyrus and fornix. And the amygdala is part of the limbic system, but it sits by itself kind of thing. You won't see it categorized in either of these lobes, these cortices usually. Sometimes it's within the hippocampal formation, sometimes it's not. The basal ganglia, whatever. It just sits out there. So all these, the limbic lobe, hippocampal formation and amygdala all make up what's known as the limbic system. Okay? And we've got very complicated, very interconnected system. So I'm just going to give you a very, very basic overview of what this thing does for it. This is a very old part of the brain. It has some major functions. Let's pick those out. Formation of memory. Formation of short-term, converting short-term into long-term memory. Okay? So, formation of new memories. Limbic system. If you have damage to the limbic system, you will often have problems in forming memories, remembering what you had for breakfast. You can remember what you had for dinner on your wedding day 30 years ago, but you do not remember what you had for breakfast. You can't form new memories. Along with that, emotions. The limbic system is very important in emotions. It's sometimes called the emotional brain. So, aggression, basic drive behavior, sexual related behavior is a function of your limbic system. Also very highly correlated or involved with the limbic system is olfaction. You can see on that diagram the olfactory bulb is included in the limbic system. And here, the olfactory bulb. There's many connections of smell within the limbic system. This is why, for example, smells are very well remembered, but they evoke emotions. You smell something you haven't smelled in 10 years, oh, that reminds me of, my mum's favourite apple pie, whatever, I haven't had for 20 years, whatever. Something like that. You remember some smell and it evokes an emotional response sometimes with certain smells. It also is important, another function of the system is controlling the autonomic nervous system. The limbic system has many inputs into the hypothalamus. And we know the hypothalamus controls the autonomic nervous system via the reticular system, via the reticular spinal tracts. So there's a lot of influence of the limbic system, of emotions, on the somatic nervous system. Okay? That's why emotional changes can affect the body, like mind over matter kind of stuff. If you're under a lot of stress, you could lose your hair, you could put on weight, you could do whatever via the somatic nerve, autonomic nervous system, and via the pituitary gland. Remember, there's a link between the hypothalamus and the pituitary gland. So the limbic system affects the hypothalamus, which affects the autonomic nervous system, which hypothalamus also affects the pituitary gland. Remember those connections through the infundibulum. So the pituitary gland can cause endocrine changes throughout the body as a result of emotions, as a result of stress, as a result of anxiety, put on weight, lose weight, etc., this kind of stuff. Okay? So the sympathetic flight or fight system is highly influenced by the limbic system. So again, emotions, memory, short-term memory, smell. For example, this is why a patient with Alzheimer's, well, let's get to that for a sec. Let's look at where this is. You can see that this is a good diagram, a good demonstration of where the limbic system is, because here it is, kind of thing, deep within the temporal lobe. And what are these things here? These what are those there? Those grey, those
ventricles.
Those are my lateral ventricles, right? The lateral ventricles. And with aging, or with Alzheimer's anyway, and other diseases, you can get what's known as periventricular grey matter atrophy. Okay, periventricular means around the ventricles. Periventricular grey matter, the brain, atrophy. This can happen in Alzheimer's. One of the early things going on in Alzheimer's is the medial temporal lobe rots away. Thiamine deficiency, B1 deficiency, we'll see again this periventricular grey matter atrophy. What I've seen, you will see in certain situations, alcoholism. Alcoholism is very well known to cause periventricular grey matter atrophy. So around the ventricles in the medial temporal lobe with alcoholics, you get atrophy of the brain. And right there is our limbic system. Right in the medial temporal lobe, the limbic system. Okay? So this is why your Alzheimer's patients can be, not all of them, but an odd one, can be very, very sweet one day. The day later you go in and she's in a very bad temper and be quite aggressive, can fall off and punch you. You know, if that's their mood that day because of, you know, the limbic system. That's why a patient with Alzheimer's can remember what they ate for their wedding day dinner, but they have no idea what they had for breakfast in the morning. Short-term memories are gone with Alzheimer's. That's one of the key signs of early dementia is loss of memory, short-term memory. The first thing to go. Sometimes this is known as, well, not that, but if it's very bad, you can get what's known as, label this, Korsakoff psychosis. This is when you've got damage to the hippocampal formation. The hippocampal formation, remember, well, is the key part of the limbic system that's involved in the production of memories, or the laying down of memories. So hippocampal formation puts short-term memories into long-term memories, lays down new memories, okay? So a patient who has damaged the hippocampal formation can't remember what they had for breakfast, whatever. Okay, so what they'll do, and in severe cases it's called Korsakoff psychosis, or amnestic confabulatory syndrome. Okay? Amnesia, can't form memory, confabulatory, they keep talking. Talk together, that means. So a patient, for example, I had one a few months ago in an assisted living centre who was an alcoholic, was anyway. And, you know, major brain damage, rotting away because of this. And I went into the guy and I said, you know, on a Monday I think it was, what did you do the weekend? He said, my son came down from Presque Isle and we went out for lunch at the weather vane. Oh yeah, what do you have? Oh, I had a great prime rib, but the vegetables were a bit soggy. I didn't really like those, but then the dessert was great. We had this, okay, great. And oh yeah, my grandson kids were there too, and they had blah 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 blah. Nice story, elaborate story about exactly what he did over the weekend. As I'm leaving the clinic, the centre, I asked one of the nurses, see, Joe's son came down the weekend, did he? And she said, no. He had no visitors the whole weekend. He had no visitors whatsoever. But he made up this massively detailed story of what he did over the weekend. It was complete rubbish. It wasn't deliberate by him. He has no short-term memory. So he fills it in. So I must have done something, didn't I? So I don't know how it came and why it came. Maybe that was a long-term memory of 10 years ago. I have no idea. But they make up this story to try to fill in the gaps, the details that basically he couldn't even remember what he had for breakfast kind of thing. But he made up this confabulatory syndrome and severe cases. Severe cases, you might see this, with damaged hippocampal formation, periventricular grey matter problems. These people with hippocampal formation, interestingly, can form procedural memory sometimes, procedural knowledge, but not declarative knowledge. You know what I mean by that? Procedural knowledge versus declarative knowledge, have you come across those terms? Huh? So declarative knowledge is something like, who was the first president of America? When was the Norman invasion of England? 1066, right? Things like that, these facts. When did the First World War start? What was the names of facts, etc., etc. Boom, boom. Declarative facts, that they're gone. You can't form those with hippocampal damage. Short-term memories. I tell you, the sky is blue. I'll give you some new fact, and you won't remember it. However, many of these patients can still have procedural knowledge. They can form new memories for procedures, how to do things, how to do motor skills. They can learn to play soccer. They can learn even to do a jigsaw puzzle. They can learn procedures. They can learn how to drive home via certain routes, whatever. They won't be able to tell you how they did it. They won't remember the fact where to turn left. They won't be saying, yeah, I need to turn left at the Citgo and then turn right onto Wilson Street. They won't know that. They couldn't tell you. Put them in a car, they can do it. They can form new procedural knowledge, but not declarative knowledge. Facts. What they had for breakfast is gone. They're perfectly intelligent, though. I mean, they're not cognitively. It's not a cognitive issue in terms of cognitive thought process. It's just they can't lay down new memories. And in the severe case, you get this Korsakoff psychosis, amnestic confabulatory syndrome. So that's the kind of memory issues associated with the hippocampal formation. With the amygdala, you can see it here. I don't know, it's all over. The amygdala is right on the front here of the hippocampal formation. Hippocampal formation, big loop comes round here, and right in front of here is the amygdala, sitting right there. There's a better picture here. You can just briefly, you can see these fiber connections here from the amygdala. This is the medial temporal lobe, right? Here's the temporal lobe. Here's the front of the medial temporal lobe. So that's where the amygdala sits. It's highly connected, you can see by these fibers, these neural fibers, to hypothalamus. And those connections are known as stria terminalis. Those are big efferent fibers, output fibers from the amygdala over and around to the hypothalamus and the septal area there. These are our drive, emotional driven fibers. This is the way the amygdala influences emotions. This is our drive related behavior, sexual behavior. I want to eat, I want to sleep, I want to drink. They come out from the amygdala over to the hypothalamus. The hypothalamus then influences somatic nervous system, etc., etc. If you damage the amygdala, as in Alzheimer's, because here's, not shown here, but here's our ventricles are right here. So damage, periventricular grey matter damage will often take out the amygdala, start eating into the amygdala. So if you get various places, if you give the amygdala e-stim, electrical stimulation, you can zap the amygdala in certain places. There's no aggression, there's no flight or fight at all. If you zap in other places, they get very, very agitated and very, very aggressive. So it depends where the damage is, but usually you're going to see if the amygdala is damaged, changes in emotional behavior. There's an interesting research in the 1930s by two Germans called Kluver and Busey. They're two blokes in the 1930s did some research on monkeys, and it kind of gives us some indication of what the amygdala and the limbic system do for you. What they did surgery, they did lesions deliberately on these poor monkeys. They cut away into the medial temporal lobe here. They gave lesions along the frontal portion of the inside of the temporal lobe. Okay? To these poor monkeys. And what happened to the monkeys? Now when you look at what changed in the monkeys based on if I lesion this part of the temporal lobe, the medial temporal lobe, they didn't know it, but they were lesioning the limbic system. What happened to these monkeys? And what they found was this Kluver-Busey syndrome. A collection of symptoms called Kluver-Busey syndrome as a result of these lesions to the medial temporal lobe, as a result of lesions to the amygdala. These monkeys were very, very tame and docile, very, very unaggressive, very quiet and relaxed, and very easygoing monkeys they were. They weren't scared of anything. They had no flight or fight response. You would put a snake into their cage and they would walk over to the snake. Normally they'd be crawling up the edge of the cage to get away from the snake. But they would go over and look at the snake and pick it up. And, what the heck's this? Chuck it away. They were very curious. Very curious. They didn't seem to remember they'd just seen something. They called it, they termed it, they named the term psychic blindness. We now term that same thing visual agnosia. They couldn't recognize what they'd just seen. Didn't care, weren't scared of it, no fear. Limbic system damage affected the emotions, affected the flight or fight system. The other thing, they were sexually adventurous, as we say. They tried to mate with anything. Males, females, or inanimate objects. Okay, I know some of you know people like that. I have friends like that. I have quite a few friends in LSU in the rugby team who had, I think, they were not afraid of anything and they tried to mate with anything. The other thing they do, my friends in Louisiana, they try to eat everything. They would, called hyperorality. They would stick stuff in their mouth constantly to try and figure out what it was. They were trying to explore the world. They couldn't, for some reason they had this sort of hyperorality. They'd explore everything. They're ceaselessly curious. They'd stick fingers in their mouth, trying to taste it, throw it away, pick something else up, pick up a snake. What is this kind of thing? They had no kind of memory of knowing what these things, that they'd just done this. They just stuck that thing in their mouth 2 seconds ago. They didn't know. They kept doing this kind of thing over and over and over again. So there are these this constellation of symptoms called Kluver-Busey syndrome that kind of give us an idea of what the limbic system does for us. You know, these Other kind of experiments have shown with monkeys have put e-stim into the limbic system, into the amygdala, and if you put it in one place, the monkey's scared stiff. They shake like crazy. Their hair stands on end. They're terrified, absolutely terrified if you e-stim a very certain part of the amygdala. Scared stiff they are, tremble in fear. Another experiment put an electrode into the amygdala, a different place, and they hooked up to a lever. Okay, and the monkey realised when he pressed that lever, they got a little e-stim injection, as it were, into the amygdala, and it hit a pleasure centre, because the monkey would just do this constantly all day long. So some pleasure centre was getting massive gratification about this e-stim into his amygdala, massively, exciting and wonderful, and the monkey would sit there, press this thing all day long, because it was, they hit a pleasure centre. So it's thought that the limbic system has these pleasure and pain centres in them that, determine our emotions. So that's the limbic system in a nutshell. Very, very brief overview. Just remember, smell, memory, emotions control your autonomic nervous system. The emotional brain, that's your limbic system, a very old part of the brain. Change the limbic system, changes inability to form short-term memories, and emotional changes, and problems perhaps with smell. Okay, that's all correlated by the limbic system. Alright, last topic for the year. This stuff. So some terms we'll throw out. Just go over some terms related to neural lesions. I don't know where I should have fit this in. I should have probably fit it in when we start talking about axons at the very beginning. But let's just go through some terminology associated with particularly peripheral nerve lesions. And I know PTs used to do this in the physical agents class, but I don't think you do. That's why I added it in. Is that right? Have you seen these terms before? Axotomesis, neuropraxia? No? I think they used to do it in physical agents. That's why I kind of threw it in here. Alright, let's look at these various terminologies for types of neural lesion, which you should be aware of. Okay, so these first three words, neuropraxia, axotomesis, neurotomesis, deal with levels of severity of a peripheral nerve lesion. Okay? Neuropraxia is the least damaging problem you can get to a peripheral nerve. This is basically a transient physiological block. It's a temporary block to the nerve transmission through the ulnar nerve, through the musculocutaneous nerve, whatever. Mildest form of neural insult is called a neuropraxia. This is things due to like a blow, a blunt trauma to the nerve, a stretch to the nerve, repetitive motion problems. So this is your brachial plexus lesion generally, hopefully. This might be a childbirth. You have a brachial plexus lesion due to a stretch injury, a tensile force, and it temporarily transmission through the nerve is gone, or at least deficit. Recovery might be minutes, might be days. Maybe a week or so if this occurred during birth, for example. But there's no lasting damage in this kind of very mild form of neural insult. The normal conduction is lost only at that site. It's stopped, there's a block there temporarily. This is your Saturday night palsy, also called crutch palsy. Another type, I'm saying something. Crutch palsy. You know how you're supposed to walk crutches, not stuck into your axilla. You get pressure on which nerve particularly? Radial nerve. You get your wrist drop symptoms, you get the elbow extension. But you take away that pressure for a while, you get rid of those crutches, you wake up, you've got your arm, you've got Saturday night palsy, you fall asleep over an armchair, and you've got this pressure into your brachial plexus from underneath. You get, minutes, hours, whatever, of problems, and then it's gone. Okay? So that's a very mild form of lesion, but it does affect neural condition. There's no change in the nerve itself. There's no damage to the nerve apart from pressure on the nerve. Then you get worse would be axonotomesis, the next level of severity of damage. Here you actually get myelin sheath damage. You might get axonal damage. But the connective tissue around the nerve is preserved. So even though you might get Wallerian degeneration, you heard that term? Wallerian sometimes called degeneration. So I damage the nerve here. I cut the nerve. The axon degenerates distal to the site of the lesion. That's Wallerian degeneration. You can see that here. The axon is gone. Wallerian degeneration. Okay? But the connective tissue sheaths are still there. Okay? And what happens is you get the Schwann cell dies off, the myelin dies off, the Schwann cells remain. So the wrappings around the axon distal to that lesion degenerate. Are basically absorbed by the Schwann cells. The wrappings go. But the Schwann cell itself remains. And they form a tube. I've got that term here. They form Schwann tubes. Or Wagner bands. Same thing. Schwann tubes. So again, I cut the nerve badly. The axon distal to the site dies off. Completely atrophies away. So obviously there's no conduction down it. It's gone. The myelin sheath degenerates. The insulation degenerates. But the Schwann cells remain. Okay, and they form into a tube called a Schwann tube. This does not occur in the central nervous system, of course. You do not get Schwann tubes in the central nervous system, unfortunately, because you've got oligodendrocytes. So it's a whole different process there, wrapping our nerves, myelinating our nerves. Okay. So once you've got that death of the axon, but you've got this tube remaining, the axon then will start to regrow. And we'll look at that in a second. And it goes down the tube and we get the innervation. It grows pretty slow. A millimetre, 2, 3, 4 millimetres a day. Something like that. So if you've got a sciatic nerve lesion or common coronary nerve lesion and the patient wants to know when will I get sensation back in my toe, it's going to be a while. A millimetre a day growing back. Okay? We'll look at regrowth in a second, a little bit more, but let's just talk about this last one here. Neurotomesis. This is the worst case scenario. Here you get complete destruction of the nerve. The axon and all surrounding structure is gone. Big old scar might develop. The prognosis is very poor for this. You're not going to get any regrowth out of this unless you have surgical intervention. Okay? There's no chance they're going to get regrowth unless you surgically reattach the end of this big gap. The amount of regrowth or the potential for that regrowth with axonotomesis will depend on the size of that gap. Okay, the size of that cut. So let's look again quickly what happens when you do cut a nerve. We cut a nerve, death of the axon distal to that. You'll also, in severe cases, you'll see death of the axon proximally to the site. And you can see what happened, the very first thing will happen in a cell body is you get the migration, or it becomes white. The colour, it becomes much paler. The cell body of a nerve that is being cut becomes very, very white. And that's called, I think I've got it here, chromatolysis, death of the colour. Okay, you get death of the colour of the cell body. And you might see what's known as retrograde degeneration. Retrograde degeneration. That is when the axon dives backwards towards the cell body, proximal to the lesion. Okay? When the axon dives down towards the effector site, it's called Wallerian or orthograde degeneration. So orthograde degeneration is the death of the axon distal to the site of the lesion, down to the skin, down to the muscle. Retrograde degeneration is death of the axon back up towards the cell body, where proximal to the lesion. Okay, so we've got this problem here, we've got a gap. We've got a Schwann tube formed. Now what happens then, a few days after the lesion, the axon from the cell body starts sending out little feelers into the gap. Pseudopodia kind of thing. Yeah, let's picture it. But what happens is the end of the axon forms a growth cone. And it sends out these Medusa-like snakes to look for the Schwann tube. So there's the end of the axon, this growth cone, and it sends out these sort of pseudopodia like this. Maybe 15 or so of these things. And they spread out and they search for a Schwann tube. If one hits it, a Schwann tube, okay, the rest will now die off. Like that. And that one pseudopodia, one growth spurt goes down the axon and all the way down to the muscle, hopefully, at 1 millimeter a day or so. Okay? Sometimes it misses. If that gap is too large, you get that thing in the middle there. This mess. So those just wrap around, those little pseudopodia, just circulate round and round looking for a Schwann tube. That Schwann tube is too far away. So they go round and round and round and make a big old mess. And that's known as a plexiform neuroma. There, this one here. Plexiform neuroma. Which can be painful. That's a sensory nerve. It's basically a stub. This is, for example, why amputees sometimes have very, very painful stubs on the end of the limb. Because all these nerves are just proliferated, going around in a big old ball, big old mess, trying to find a Schwann tube when there isn't one there, and therefore they ball themselves up into this plexiform neuroma, which can be very painful. You can get this situation on the bottom here. Anomalous reinnervation. You can see that there, where 2 axons have been cut, and they've cross-switched. They've switched to the wrong one when they're trying to grow to find their own Schwann tube. And you can see that term here. Anomalous reinnervation. You get some interesting things going on there, where you try to blink your eyelid or something, then you twitch a muscle in the wrong place. The muscle is reinnervated by a different neuron. Okay? That's anomalous reinnervation. If that gap is too long, as I said before, too large, you're not going to get it growing back. This is why you need surgical intervention. You go and join those ends up closer so there's a better chance of those neurons, those axons crossing over. I think I saw some research in England where they actually now they're putting a little bit of muscle tissue. They cut a little bit of muscle tissue with sort of grooves in it, right? Stick it in the gap, and that improves the likelihood of that axon growing through the muscle and hitting its correct bunger or Schwann tube to be able to get correct reinnervation. Again, if you have a central nervous system problem, you don't get this. You cannot get this regrowth of axons down Schwann tubes. That's why your spinal cord injury nerve patients, spinal cord injury, your brain lesion, etc., that's gone. You do not get regrowth of those axons down those tubes. One thing you will find sometimes with a neural lesion, like a cut in a peripheral nerve and a motor nerve, remember these two terms. Fasciculations and fibrillations. Remember those? When you see those, what kind of disease, generic group of diseases would you see fasciculations and fibrillations? That's your lower motor neuron disease, right? And what happens there, you can see you've got some kind of a lesion, like whatever. What's what I'm thinking about? What's the most common peripheral healing brain? Healing brain. You've got a lesion here. What happens is at the site of the lesion, signals are sent out spontaneously, sort of out of the lesion site, down the neuron. And as it goes down this one alpha motor neuron, it's going to innervate all the fibers. It's going to contract all those fibers innervated by that motor neuron, that motor unit. And therefore you see a fasciculation. The whole motor unit contracts. You see what's known as a fasciculation. Those are twitches you can get, those myogenic fasciculations. You see, don't you, some of your eyes start twitching, or one of your muscles in your forearm. You've had that where it just twitches uncontrollably. And you have to whack it, you punch it, and it keeps going. What the heck's going on, And it twitches. That's a fasciculation. Okay? However, if the lesion is down here more, or the degeneration as the nerve dies back, down to where the nerve is branching, you don't get a coordinated contraction of all those muscle fibers within a motor unit. You get a few of them will fire by themselves. And that's your fibrillation. Single fibers firing by themselves are fibrillations. You cannot see that by the naked eye. You can see that with the electromyography. You cannot see it by the naked eye. And those are two symptoms of a lower motor neuron lesion. What else have we got to talk about? Anything else on here? Last word, very last word then of this semester. You do see recovery of function with a central nervous system lesion. If you didn't, there'd be no jobs for you. Okay? So there is recovery of function after a stroke, isn't there? They do get better generally. Or a spinal cord injury or traumatic brain injury. People, with our help, improve function, hopefully. How do they do that? Obviously it's your skill, right? What is going on underneath the, at the central nervous system level, given that A, we cannot regrow axons in the central nervous system, we cannot bud off new cells anywhere in the nervous system apart from the olfactory system. So how is it people are getting better with your lot's help? There's a few theories, and those are basically vicariation, redundancy, and diathesis. These are three things that help you look like a good therapist. Okay? Vicariation basically means other parts of the nerves, other nerves take over for the ones that are damaged, in essence. So this part of the brain is damaged, so this part of the brain will take over and do that function for the person. Okay? Vicarious, you watch somebody, vicarious arousal, whatever. Vicariation is where basically the slack is taken up by other nerves. Okay? They see there's a problem, and therefore this part of the motor cortex, takes over for that lost area. Redundancy, which I don't really believe, but there's some evidence for this, is this theory that you've got a backup system for everything in the body anyway. We have these supposedly dormant fibers waiting to for somebody to have a stroke, and then they take over. You've got redundant systems. So vicariation says other systems that weren't involved, were never designed to do this function for you. Redundancy suggests, and I have a hard time believing this, but it suggests that we have backups. It's waiting to be activated when something gets damaged. Yeah, I don't know about that one. We want to waste time to have a backup nerve system, backup everywhere in case something gets unnerving. But definitely you see this diasthesis problem. But this is some of the recovery you will see in your patients after a stroke as a result of removal of diasthesis. So what this is, basically, diasthesis is a spinal shock, a shock to the nervous system, which gives you lots of symptoms. In other words, I have a small stroke here, or have a small spinal cord injury here. But what happens when you injure the nervous system, the whole nervous system goes into shock. It freaks out. Okay? It all goes haywire. And your symptoms you'll see in your patient after a stroke are due to this massive spinal shock, due to diasthesis. Okay? But that's not really the lesion. That's just a shock to the system. Now that, you gradually recover from that. The shock dies off, dies away. And you're left with the symptoms that the lesion itself caused. I cut this part of the spinal cord, therefore I've lost that specific function to that little finger, that arm, that face, that leg, whatever. So when your patient comes in after a stroke, after spinal cord injury, after whatever, they're probably suffering a lot of symptoms you'll see are due to diasthesis. And then you start treating them, and lo and behold, that shock to the system dies away, gradually resolves itself, and the patient looks like they're improved. They are. They're getting a lot better, and obviously that's due to you, your treatment. Not to the fact that this is resolving itself, it's the fact you're great therapists. And you claim that as, your treatment approach. But the fact is, that spontaneous recovery you'll see in early stroke patients, whatever, they gradually get better. Some of it is going to be due to the natural resolution of this shock to the system that injury gave them. And now the residual symptoms they've got at the end, those permanent sort of symptoms, are due to that lesion itself. Then your treatment starts working and working and working, hoping you're getting some vicariation, hoping you're getting adapting, and then you look at compensation and whatever, various treatment approaches, how early you let the patient compensate, or do you push for normal movement patterns, etc., etc., etc. That's a whole different ball game. That's all your treatment. Unfortunately, I can't talk about that this semester. Okay, we're done. Questions on any of this stuff? Review the whole semester, right? Okay, so I'll be around all this week, next week, for questions, emails, or whatever.